mitochondria, and I'll say more about this later. 2 is invoking specialized enzymes, and I mentioned the flavoproteins earlier. Those play a critical role. They are able to select hydrogen over deuterium in their reaction. They have a very sophisticated mechanism, which is related to proton tunneling. It's a biophysical mechanism that allows them to be sure that their product contains protons instead of deuterons for the most part when they perform their reactions. They refuse to react if the hydrogen at the position they're trying to yank it off is a deuterium instead of a hydrogen. Fascinating paper, this paper from 1964. Back in the 1960s, they did some experiments with deuterium, heavy deuterium water. They were able to make this water for the first time around that time, and they were curious about what would happen if they fed it to rats. Or mice. These mice received 5% dextrose solution in pure D2O water, 5% in a dextrose water solution. And their appetites were voracious. They immediately started being very hungry, and they also became very angry and irritable, and they were savagely attacking each other, became fierce. And then at varying intervals, and this was only after a few days, the animals became hypoactive, lethargic, and dramatically weak. They basically lay down and let the other animals that were still energetic kill them. And all of the animals died within 6 to 10 days. So this stuff, which just tastes a lot like water, is incredibly toxic if you drink it straight up. Now, it's interesting to me that in Russia has really been a leader in this whole idea of deuterium. And it came about because there were people up north in Siberia who had who were very, very healthy and had a very long lifespan, despite being in an area where it's so cold and winters are so horrible that you can barely have a growing season. They ate really a limited diet because of this. But their water came from glaciers, and the water was deuterium depleted. And it took them a long, long time to figure out that it was, in fact, the water that appears to be a major factor in their good health. And so this is how Russian scientists became interested in deuterium, and have also learned how to make deuterium depleted water, which you can now obtain on the internet as a resource to help maintain good health. So this is manufactured in the laboratory, deuterium depleted water, but the glacier water is naturally deuterium depleted, and places that have glacier water as their source of water tend to have healthy lives. And so deuterium depletion maintains strong hydrogen bond networks in DNA, which is to say that DNA can become unstable if there's too much deuterium. And this can inhibit tumor progression. This is a paper by Laszlo Boros and others that was actually a very seminal paper for me. Laszlo introduced me to deuterium in December 2019, and I have been deeply studying it since then because I believe it is very, very important. Interestingly, a ketogenic diet is a deuterium depleted diet because it's been shown that fats in particular, like bacon, butter, lard, these foods are significantly deuterium depleted compared to other foods. So glyphosate, what is glyphosate? It's the active ingredient in the herbicide Roundup. It's pervasive in our food supply, especially in America. We have 4% of the world's population, and 20% of the world's glyphosate is consumed here. The manufacturer claims it's very safe for humans, but I don't think that's true. And my research has shown, in fact, other people's research as well have shown that that's not true. It kills weeds by suppressing an enzyme in the shikimate pathway. The enzyme is called EPSP synthase. The argument is our human cells don't have that pathway, don't have that enzyme. There's no way glyphosate could affect us. But the problem is that our gut microbes use that pathway to produce essential amino acids for the host. We can't make those because we don't have a pathway. And those essential aromatic amino acids are precursors to all kinds of really important biologically active molecules. So there's strong correlations between the rise in glyphosate usage in the United States and the rise in multiple neurological, oncological, autoimmune, metabolic diseases. And this paper by Swanson et al. Has like 30 diagrams of plots over time of various diseases that are going up exactly in step with the rise in glyphosate usage in the United States. So sulfate is the other thing that I've been focused on for ever since really the beginning when I first started looking into metabolism and disease. I was particularly interested in autism, and I zeroed in on sulfate deficiency or sulfate mismanagement as a key factor in autism and other diseases. One of the things sulfate does is it maintains this gelled water that I mentioned earlier, which is also known as exclusion zone water. Gerald Pollack has made this topic popular among the general people because he's been able to publish books that make this information accessible to someone who isn't an expert. Really fascinating stuff. This glycocalyx is the is this complicated structure shown here schematically that aligns the blood vessels. And this is the wall of the endothelial cell, and then this is the inside of the cell over here. And the membrane, of course, has these two, this lipid membrane, it's a bilipid membrane with these polar lipid molecules arranged this way with a hydrophobic part inside the membrane and all kinds of different things connecting through. But what I think happens is that the glycocalyx traps the deuterium, and Pollack has shown that the glycocalyx releases protons, and those protons are going to be deuterium depleted, and then they travel past the membrane into the cytoskeleton, and then they're able to deliver deuterium depleted protons to the mitochondria as well as to the lysosomes, and both of those, that's critical for both of them to be supplied with this healthy water. And so sulfate is critical for maintaining the geled water. And then these sulfated glycosaminoglycans, which is this glycocalyx, they become depleted in sulfate when they are chronically exposed to glyphosate and probably to other chemicals as well. So F-actin is a very, very interesting molecule that I've been studying. Actin has these two forms, this kind of independent form here, G-actin, or it can be organized into these fibers called F-actin. And these fibers have very interesting properties. This is a protein, but the water around that protein becomes what they call hypermobile. It's very fluid water that's created around the boundaries of this protein. This protein makes up a lot of the cytoskeleton. And so it's maintaining fluid water, which is where these protons can travel along those fluid water pathways in order to get into the mitochondria. And the mitochondria actually assemble F-actin along their outer membrane when they're exposed to toxic chemicals that inhibit the respiratory chain. And certainly glyphosate is a toxic chemical that inhibits the respiratory chain. And so my suspicion is that this allows them to pull in extra healthy protons in from the outside world via this mechanism. So NAD is essential to support mitochondria. What is NAD? Nicotinamide adenine dinucleotide. It's an essential cofactor in oxidation reduction reactions, which of course are a major part of metabolism. It exists in four forms, which is basically with and without the hydrogens, that's the H, or with and without the phosphate, that's the P. So it's represented as NAD+, NADH, NADP+, and NADPH. All of these, you see them in all kinds of equations for metabolic metabolism. And the H that you see here is a very special H because all the enzymes that deal with putting this H here are good at selecting protons over deuterons. And so the mitochondria supply the energy in the form of ATP. They depend critically on a proton ATPase pump to make the ATP. And the protons derived from NADH and NADPH are pumped into the intramembrane space, and then they flow back out through the pumps. Deuterium gums up the pumps. It's sort of like putting sugar in the gas tank. And so here's a mitochondria, just to get you the picture of it. These are little organelles. There's lots of them inside the cells, especially the muscles and the neurons. This is the intramembrane space around the light blue part around the edges. The green is the matrix, and all the DNA and stuff is, and lots of things are in the matrix. The protons are pumped into this intramembrane space, and then they come back out at these ATPase pumps, which are these little dots that are pushing. They're making the ATP using the energy of the protons coming back out again to create the ATP. So now this is a picture of the electron transport chain. This is the intramembrane space. This is the cytoplasm. So you have this inside the matrix over here is where all the action is taking place. And so you have NADH losing its H and putting it in there, NADH2 losing its two H's. So all these H's are going into here. And these guys are all low deuterium protons. So these are all low deuterium protons inside. And then you get basically the H's all go in, that's the protons, and then they come back out. They gather and they come back out. And as they move out, that motive force is what turns ADP into ATP, adding one phosphate, and also turns oxygen into water, adding two hydrogens. So in fact, four hydrogens to make two water molecules out of two oxygen atoms. So this water is going to be deuterium depleted water, because it's coming from those protons that are deuterium depleted. And this is crucial. This is absolutely crucial for the mitochondria to work properly. So here's a picture, sort of more realistic schematic, where you've got this cytoplasm out here. This is inside the mitochondrial matrix. This is one of those little wiggles in the intramembrane space over here. all the protons are being pumped in, and then they gather and they come back out over and they get concentrated over here. And then you have these ATPase pumps lined up, ready to go. As the protons come back out, they produce ATP. And then if you've got deuterons in here, they're going to basically wreck the pumps, and the pumps will start spewing out reactive oxygen species and make less ATP. And that's absolutely crucial, because that's what's happening with the mitochondria, oxidative damage due to mitochondria misbehaving, not performing well, being sick. This is happening because of all that deuterium that's in there. So glyphosate, glycine, and flavoproteins, my next topic, the big picture. Flavoproteins are an important class of proteins that transfer hydrogens to and from NADPH using flavins, FAD, FMN, as intermediaries. Now, I want to say that all of these guys, the NAD, the FAD, the FMN, they all contain phosphates, multiple phosphates. And that's important because glyphosate disrupts phosphate binding in enzymes. And that's where I'm going to get to in a moment. So its disruption of the shikimate pathway leads to deficiencies in tryptophan. Tryptophan is one of the aromatic amino acids that comes out of the pathway. And tryptophan is a precursor to NAD. So that you're going to have NAD deficiency if your microbes are not able to make enough tryptophan. And the same thing is true for the flavins, the FAD, riboflavin. So you have these two B vitamins, niacin and riboflavin, that are crucial for making these critical molecules that are going to be disrupted because of glyphosate disrupting the gut microbiome. Tryptophan is also a precursor to serotonin, which is the feel-good hormone, and serotonin deficiency is linked to depression, obesity, and violent behavior. And both glyphosate and excess deuterium are going to cause a depletion of serotonin, and that's going to cause these problems. My research strongly suggests that glyphosate suppresses enzymes that bind phosphate, and you can read about that in this paper that I collaborated with Sarath Gunatulake on this paper, where we talk about that. In fact, particularly with this, with the enzyme that it disrupts in the shikimate pathway, EPSP synthase, that is a situation where glyphosate disrupts phosphate binding. That's been shown in many studies. It disrupts the phosphate binding of the substrate PEP. And I generalize that to multiple other enzymes that bind phosphate in my theory. And so these guys all contain multiple phosphates. Flavoproteins contain essential glycine residues at all the sites where they bind phosphate. And so does EPSP synthase. So there's tremendous susceptibility to glyphosate's mischief through glycine substitution. And here's my hypothesis. Glyphosate disrupts proteins that bind phosphate. Well known, it kills plants. It kills all plants by suppressing EPSP synthase, except for those that have been engineered to resist it through GMO technology. And it also kills many microbes for the same reason. It blocks EPSP synthase binding to phosphate in PEP. I've said that already. Glyphosate is a glycine molecule. It just has an extra unit attached to its nitrogen atom, which is a methylphosphonate unit. And down here is a methylphosphonate. This thing's a methylphosphonate bound to sodium, and this is a phosphate bound to sodium. And you can see that these two molecules are very, very similar in shape and size. So this one is replacing the place where this one is supposed to go and preventing the substrate from getting close to the enzyme and therefore messing up the reaction. And this is happening in multiple enzymes throughout the body and throughout all life, I should say, all life forms. So in the enzyme that it famously disrupts, the binding site for PEP has a highly conserved glycine residue. If you take that glycine out, if you change the genetic code so that it codes for alanine instead of glycine, which is one extra methyl group, you completely kill glyphosate's ability to disrupt that enzyme. And that's because the code is changed. It's the code that glyphosate is matching to stick the glyphosate residue into the spot where the glycine is supposed to go during protein synthesis, messes up the protein's function. And this is the basis for any GMO glyphosate resistant crops. They change the code so that it's alanine instead, or you find an enzyme that has alanine instead of glycine, a version of the enzyme from a microbe, put that into the plant, and the plant is resistant to glyphosate. So this is really quite interesting to look at some of these proteins, these flavoproteins. So there's a class A flavoprotein called monooxygenases, and these are very important enzymes that do many things, but their main function is to add molecular oxygen to what's called lipophilic compounds that don't easily dissolve in water, and they're often toxic, xenobiotics. And then by doing this, it makes them more soluble to ensure they can be excreted through the urine. And so these enzymes have a particular pattern in them with this Rossmann fold, this DG unit, and this GD unit, two GD, a DG, a GD, and a Rossmann fold are critical pieces of these enzymes that help them perform their action. And you can see that there's 3 glycines here, there's 1 glycine here, and there's 1 glycine here. So all of these glycines are susceptible to glyphosate substitution to disrupt the function of these enzymes, resulting in an inability to detoxify xenobiotics in the liver. Now, this is another paper that talks again about some glycines. This is a motif, a GXXXG motif, which stabilizes helix beta strand interaction. These things turn out to be very, very important. And then there's another motif here, which is either 2 wild cards spacing 3 glycines, or you could have just 1 here, GXGXXG. The wild cards means it could be any amino acid. And so these are motifs that stabilize the binding sites of these enzymes that have these so-called Rossmann folds. So this is all very basic biophysics, maybe biochemistry, concerned with enzymatic action, where they've seen that these glycines are absolutely critical for these proteins to function properly. And so these motifs stabilize FAD or NAD binding, Rossmann folds, and binding of the nucleotide cofactor to the domain. So that's absolutely critical for the protein to work. And you can actually find proteins that are flavoproteins. You can find novel flavoproteins by using these patterns to search the protein literature. So this is an article that shows that Roundup, which is glyphosate, is the active ingredient in Roundup. Roundup suppresses NADPH reductase. NADPH reductase is a very important enzyme for pulling NADP plus back to NADPH, so it can do it again. So NADPH, is supplying these H's, which are these deuterium depleted hydrogens, to all kinds of substrates. And you need to get the H back, and you need to do it using an enzyme that's able to make sure it's H and not D. And that's what these flavoproteins do. They're very, very special to be cleverly designed. They're using that FAD to help them do that, the flavin, so that helps them to produce a version of NADPH that has a quality assured H there. And so this paper talks about aromatase. Aromatase is an enzyme that converts testosterone to estrogen. And so it really disrupts development when aromatase is broken. And it's been shown, in fact, that glyphosate causes excess testosterone during development, which is linked to problems later on in life for the offspring that are exposed to too much testosterone in utero. But this enzyme, aromatase, depends upon pulling this NADPH back in order to work. And when that is broken, then you can't use that enzyme properly. And so this experiment just showed that glyphosate, Roundup, suppressed that enzyme. And so I've gone through the research literature and found all kinds of papers that talk about various enzymes that glyphosate has been observed to, just experimentally observed to suppress. And many of them are, in fact, enzymes that bind to these various phosphate-containing nucleotides. And so these are just some of the others and you can read about it here in this book, Toxic Legacy, which will be available July 1. Besides NADPH reductase, other phosphate-binding enzymes that glyphosate's been shown to suppress experimentally include succinate dehydrogenase. We're going to get to that in a moment. Aromatase, cytochrome P450 enzymes, those are essential in the liver for many functions. One is to produce bile acids, activate vitamin D, many functions. Those are getting disrupted. NAD transhydrogenase, which transfers hydrogen from NAD to NADP in the mitochondria, also very, very important for maintaining the mitochondrial health. G6PD, which restores NADPH from NADP+, that's also very, very critical. And then Rubisco, which is the most common enzyme in the world in plants, and it's essential for plants to be able to grab carbon dioxide out of the air and turn it into organic carbon. So that could be playing a role in climate change. So all these enzymes are being affected by glyphosate. So succinate dehydrogenase, I want to go into detail with this enzyme because it's a very nice example of many of the effects that are critical, I think, for many of the effects that are happening and very much linked to cancer. So here is a paper, The Role of Mitochondrial Dysfunction in Cancer Progression. And so this paper is showing that defects in mitochondrial enzymes, and this includes succinate dehydrogenase, fumarate, hydratase, isocitrate dehydrogenase. These are all enzymes that are likely to be affected by glyphosate. And it's associated with both familiar and sporadic forms of cancer. And then the mitochondrial dysfunction causes pyruvate dehydrogenase to leave the mitochondria and go into the nucleus and then produce acetyl-CoA enzyme A there instead of in the mitochondria. It's very, very interesting that that's what happens because that's so clever that not only does that prevent you, it sort of shuts down the mitochondria because you're not getting this acetyl-CoA, but instead it's delivering it to the nucleus where it can then start acetylating these histones. So histone acetylation, these are the histones keeping the DNA locked up. That the acetyl-CoA comes in and puts the acetates on these histones, opening them up and then activating the DNA there to start making proteins. So that's how you can turn on different genes to express different proteins. And this happens when with the stress, when we have the mitochondrial stress with these enzymes not working, then this is what happens. And it's just very clever, but it's going to start turning on oncogenes that are going to start looking like cancer. So here's succinate dehydrogenase, more about it. It's basically the complex 2 of the mitochondrial chain. It's the only enzyme in the mitochondria that's intimately involved in both the citric acid cycle and oxidative phosphorylation, which are 2 major roles of the mitochondria. It is a flavoprotein, so that means it's going to be susceptible to glyphosate. And it extracts 2 protons from succinate and puts them into the membrane by working with coenzyme Q10. So coenzyme Q10 is something many people are recommending as supplements to help support the mitochondria. You need this succinate dehydrogenase to be working properly in order for coenzyme Q10 to work properly, because it's going to hold those 2 protons in the membrane. And then later on, they're going to be pumped into the inner membrane space to be supplying the protons that the ATPase pumps need to be able to do their work. And so there's many genetic mutations in succinate dehydrogenase, and they're associated with many cancers. And you can read about that here: neuroblastoma, breast cancer, colon cancer, renal cancer, melanoma, uterine cancer, prostate cancer, endometrial cancer, bladder cancer, gastrointestinal stromal tumors. So all of those are linked to genetic mutations in succinate dehydrogenase. So glyphosate suppresses succinate dehydrogenase. This is a specific study on E. coli. They looked at different proteins that were suppressed by glyphosate. They found a long list, but in particular, they found 3 different components of succinate dehydrogenase complex that were suppressed by a factor of 3 to 4-fold in these E. coli microbes. Another study on rat liver mitochondria operating in vitro in isolation, and they exposed them to Roundup or glyphosate alone. They found that Roundup, but not glyphosate, suppressed succinate dehydrogenase, and they suspected that the glyphosate by itself was unable to get into the cells, into the mitochondrial membranes, and so it didn't have an effect. But the Roundup enabled caused additional toxicity, and in fact, it's been shown that the extra adjuvants that are added to Roundup make the glyphosate much more toxic, and they are toxic in and of themselves. Experiments have shown that. And so the Roundup formulation is much more toxic than glyphosate by itself, in part because it allows glyphosate to get inside, but in part because those things are messing up the membranes of the cells and causing damage on their own. Analysis of the mechanism of glyphosate suppression suggested that it disrupted the binding of succinate dehydrogenase to FAD. So this is specifically what I've been saying, and that's what they showed experimentally. It messes up succinate's ability, dehydrogenase's ability to bind to FAD by filling up the space where the phosphate is supposed to go. So here's just these are lovely images that you can get from this website, and this is a no, it's not Unipro, something else. But I found this, it's really lovely on the web where you can do these searches and you can look for specific proteins and find out if they have binding to these nucleotide units. This is FAD, NAD. So these are the guys we've been talking about on the left here. And then in here are the places where binding happens. And this is one of these GXGXG sequences here, but it's got 4 glycines because one of the wild cards is also a glycine. So 4 glycines in the specific succinate dehydrogenase enzyme at this place. This is the glycine here. And you can see that all this binding is happening around these glycines to this unit, the double phosphate unit in the FADH. This is the FADH molecule. It's got these 2 phosphates. And where it binds the phosphate right here, which is right where these glycines are. So all of this is not going to work if there's glyphosate substituting for any of these glycines in here. And then this is a paper that again shows key mechanism of this effect was through suppression of succinate dehydrogenase. So this is yet another paper showing that glyphosate suppresses it. It induced apoptosis and necrosis in human umbilical, embryonic, and placental cells. So this was an in vitro study showing damage due to the succinate dehydrogenase, which is this SD right here, being messed up by glyphosate and causing the cells to commit suicide. How excess deuterium induces a proliferation when there's too much deuterium in the mitochondria, which is you're going to get too much reactive oxygen species. And then the reactive oxygen species do damage. One of the things they do is they spontaneously convert oxaloacetate into malonate. And malonate is a very, very interesting molecule which suppresses succinate dehydrogenase, blocking the citric acid cycle. And now you get this pathway towards cancer. And the cancer cells adopt a pathway that pulls the oxaloacetate out of the TCA cycle to make aspartate in the cytoplasm, short-circuiting the cycle. And then the aspartate can be used to make nucleotides and proteins that are needed for proliferation. So it basically shuts down the mitochondria, starts pushing things out that can build up nutrients that can supply the building they need to make the extra DNA and the extra membranes to be able to clone themselves. So it makes the cancer become proliferative. And here's a schematic of this. Malonate is produced spontaneously from oxaloacetate via reactive oxygen species, which are reduced when you have too much deuterium. It suppresses the succinate dehydrogenase. It shuts down ATP production. And the cancer cells then take the oxaloacetate, convert it to aspartate, and then use the aspartate to make the tools that they need. So here's the oxaloacetate getting oxidized, suppressing succinate dehydrogenase, blocking the pathway, and then instead pyruvate comes through a different pathway to produce aspartate. And this is just another more complicated version of the same thing, basically. But it's quite interesting because the mitochondria shut down the ATP cycle, then they have to make ATP elsewhere. So they do it through glycolysis. They make tons and tons of ATP by turning sugar into lactate. They go from glucose to pyruvate to lactate. But if the lactate branch, I guess we're going to get into that later. Sorry, I'm getting ahead of myself. So they, and then they just use the mitochondria to turn out nutrients rather than burning them up. They basically do the reverse cycle that allows them to get nutrients that can build the DNA and the proteins that they need. And so basically you've got coming out of the citrate coming out, getting converted to aspartate on this pathway or getting converted to acetyl-coenzyme A on this pathway. They use this to make the lipids and they use this to make the nucleotides and the proteins. And then they've got all kinds of resources to be able to clone themselves. So lipid pathways in cancer, the big picture, the synthesis of long-chain polyunsaturated acids involves a series of reactions catalyzed by elongases and desaturases. Longer chains in the membrane protect from hydrogen leaks. So you want longer chains. The membrane lipids are released under stress conditions, and then they get processed through three primary pathways. And so there's a lipoxygenase-based pathway, cyclooxygenase pathway, and a CYP enzyme-based pathway. Each of them produces a different set of complicated molecules that your eyes glaze over when you try to read these papers: leukotrienes, prostaglandins, and cannabinoids. And this one is an interesting path that looks very productive. The other two cause inflammation. So you want to go this way, but you can't because the CYP enzymes are being suppressed by glyphosate. And the diverse products of these modifications are powerful signaling molecules, these leukotrienes and prostaglandins, powerful signaling molecules that cause inflammatory responses. And these pathways are geared towards producing deuterium depleted water, which I think might be the primary product. And this is something people are overlooking. They see water as a byproduct. They hardly notice that there's water being produced, but that's the critical product. And then glyphosate can be argued that it would disrupt both of these pathways. This one's actually pretty hardy against glyphosate, which is why I think it's been showing up a lot as a problem in metabolism in various people who are sick. And so here's a pathways of fatty acid synthesis and metabolism. You can see this is linoleic acid, and then you get down to arachidonic acid, and you get down to these prostaglandins, leukotrienes. And over on this side, you have the omega-3 fatty acids. This is omega-6, omega-3, and they go in to produce this EPA and DHA. These are sort of the highly unsaturated omega-3 fatty acids that people have been taking as supplements. And so you have all these different metabolites that are showing up. And so this D6D and D5D, I'm going to take a look at them in a little more detail here in a moment. Highly unsaturated fatty acids are synthesized using D5D and D6D. And this is a mechanism to renew NAD+. in situations where the mitochondria are broken, because normally the mitochondria take out the H of NADH. NADH is produced through glycolysis. So when you burn a lot of glucose, you make a lot of NADH. You need to get it back to NAD+. You normally do that in the mitochondria. And the whole point is to deliver this H to the mitochondria so you can supply them with deuterium depleted water, deuterium depleted protons. And so when you have a problem with mitochondria, one thing they can do is to grow long-chain fatty acids from short-chain fatty acids. They add new units and then they dehydrogenate them. So it's 2 steps. And these dehydrogenases, D5D and D6D, will reproduce NAD+ and help to restore the balance, because you've been making all this NADH with glycolysis. And so basically what you have here is this oxygen going to H2O. This is actually 2 water molecules from oxygen in this reaction with D5D and D6D. It's making this deuterium depleted water and through these enzymes and feeding it to the mitochondria. So D5D and D6D are desaturases. When the electron transport chain is sick because there's too much deuterium, the cell naturally reverts to creating fats that are more highly unsaturated by extracting the hydrogen in the fats to make DDW and repair the mitochondria. And then glyphosate may be interfering with this process as well, because these enzymes bind flavins. So here's a reaction for these desaturases. There's a polyunsaturated fatty acid here. You need some iron, some protons, oxygen, pure oxygen. And what you come out with is a highly unsaturated fatty acid, oxidized iron, and 2 molecules of deuterium depleted water. So these things produce these HUFAs from PUFAs by pulling out more hydrogens, putting those hydrogens, adding them to water, oxygen to make water, and you get your DDW out of this reaction. So and at the same time, you're making these long and polyunsaturated fatty acids, which will help to protect the mitochondrial membrane from proton leaks. So inflammation, cancer, and oxidative lipoxygenase activity are intimately linked. Lipoxygenases oxidize arachidonic acid and its derivatives, producing leukotrienes, which are pro-inflammatory, and ultimately lipoxins, which are anti-inflammatory. And leukotrienes activate this NF-kappa B pathway, TNF-alpha expression, inflammatory signals. So 15-LOX is highly expressed in white blood cells, and overexpression of these lipoxygenases has been implicated in many human acute and chronic inflammatory diseases, such as asthma, atherosclerosis, rheumatoid arthritis, inflammatory bowel, dermatitis, and cancer. And the leukotriene LTB4 is a potential stimulator for cancer cell growth and also plays a role in the formation of reactive oxygen species in response to hypoxia. So here's another equation for the cytochrome P450 enzymes, which this one metabolizes arachidonic acid. And it starts with oxygen and then produces 1 water molecule plus 1 proton with 1 oxygen. But the interesting thing about this reaction is that the product is an endogenous cannabinoid, which stimulates the cannabinoid receptors just like THC. So CYP enzymes are suppressed by glyphosate, and that's going to interfere with the ability for arachidonic acid to be turned into this endogenous cannabinoid, which is going to then end up with a deficiency in the cannabinoid, but also the arachidonic acid is going to go down another path that's going to cause inflammation. So the endocannabinoid system is a target for cancer treatment. The cannabinoid receptor agonists inhibit cancer cell proliferation through various receptor-mediated mechanisms. So you have, it's been shown that cannabinoids can treat many types of cancer and suppress metastasis, suppress angiogenesis. And then this was a study that showed that HER2 overexpressing breast cancer cells arrested, CBD arrested cancer cell proliferation by inhibiting these signaling molecules. And then it locks the cell cycle at this arrested at S phase, prevents it from falling through. So anti-tumor properties have also been seen in pancreatic cancer. So basically, natural endogenous endocannabinoids would be doing this if they were working, but the CYP enzymes are being disrupted by glyphosate. So preclinical and clinical studies, ketogenic diet and cancer therapy. This was done on rodents, and they showed, they basically showed the green is where these things were successful. So they were giving them a high-fat diet, and And they found anti-tumor effect is the green. So you can see there's a lot of green here. There's some bad news for kidney cancer here. Melanoma doesn't look so good, but most of the cancers look like it's more of a win than a loss to use this ketogenic diet in this experiment. Hydrogen gas for cancer therapy, clinical trial involved 58 patients with advanced non-small cell lung cancer. And they used all kinds of different delivery mechanisms, drinking hydrogen dissolved water. It's non-toxic, it's inexpensive. So there's many, many benefits of hydrogen gas therapy. And the interesting thing is that hydrogen gas is naturally deuterium depleted because deuterium tends to stay in the liquid phase. And in fact, this study showed, this was back in 1961, they showed that microbes produce hydrogen gas that's incredibly depleted in deuterium. This was down to the level of 30 parts per million, which is extraordinary. The hydrogen gas that was produced by these cells, regardless of whether they use glucose or fumarate as the substrate to make the gas. So I think these enzymes are able to specialize in making sure that a product is deuterium depleted, but also just the natural process of evaporation is going to encourage low deuterium in the hydrogen gas. And so hydrogen gas in cancer treatment, naturally generated by hydrogenases, rapidly diffuses through cell membranes. It suppresses glioblastoma. It suppressed all kinds of aspects of the cell growth. It inhibited some pathways that are known to cause proliferation, lower incidence of renal cell carcinoma, suppression of tumor growth. A rat model of NASH, hydrogen-rich water, inhibited inflammatory cytokines. So all of this is good news for the hydrogen gas. Microalgae is another thing that's been used in cancer therapy. They're rich in these long-chain PUFA and docosahexaenoic acid, DHA, which accumulates in cardiolipin in the mitochondrial membrane and then causes lipid peroxidation, and which produces DDW, but also causes these cancer cells to shut down. They're also rich in fucoidin, which is a fructose-containing sulfated polysaccharide that shows anti-proliferative, anti-angiogenic, anti-tumor impact in both lung cancer and melanoma. And the benefits were proportional to the number of sulfate groups. So this really gives you the hint that sulfate is a feature. Sulfate is a has therapeutic value in cancer to help sequester deuterium in the gelled water. And here's deuterium depleted water therapy for terminal cancer. 2 studies, one on lung cancer metastasized to the brain and one on stage 4 pancreatic cancer. Both cases, standard chemotherapy was augmented with DDW. And in both cases, they saw significant improvement in survival time. 56 patients who consumed DDW had 20 months versus only 6 months for the patients with chemotherapy alone. And this one also showed an exceptionally long survival time for patients with metastatic brain cancer. And there's 2 more studies that you can read about showing benefit of deuterium depleted water on cancer therapy. How to minimize cancer risk. Big picture, eating a certified organic diet consisting mostly of whole foods may be the best strategy. Sulfur-rich foods, which can assure adequate supplies of glutathione for antioxidant defenses and sulfated GAGs for the glycocalyx. Foods rich in riboflavin and niacin, which will support sufficient levels of NAD and FAD. Foods rich in polyphenols, flavonoids, terpenoids help maintain low deuterium in the mitochondria. A high-fat diet is a low deuterium diet, and then drink glacier water or synthesized DDW. And so here are some foods, levels of deuterium in various foods. So flour, this is going from low to high. So flour is the worst, sugar a little bit better, cottage cheese coming down to 136, olive oil 130, then you get butter 124. Organic butter is lower than non-organic butter, ghee 112, and then coconut oil 110. So these fats are all very low in deuterium compared to these sugar-based foods. Riboflavin, and so here's some sources of riboflavin in order to make the flavoproteins, the FAD for the flavoproteins. And that includes, beef, tofu, milk, fish, mushrooms, pork, spinach, almonds, avocados, and eggs. Foods high in niacin, especially animal products like meat, fish, and poultry. Vegetarian sources include avocado, peanuts, whole grains, mushrooms, green peas, and potatoes. G6PD and fatty acids, glucose-6-phosphate dehydrogenase converts NADP plus to NADPH and is able to favor H over D. NADPH is the largest source of hydrogen and fatty acid biosynthesis, and that's how you get this low deuterium in the fatty acids. And so E. coli and Bacillus subtilis produce deuterium depleted fatty acids due to their high dependency on G6PD to restore the H. And so algae, like chlorella, they produce highly deuterium depleted fatty acids, which becomes then a very good source of a low deuterium nutrient. Natural phenolic compounds from medicinal herbs, and dietary plants, phenolic acids, all these different complicated molecules that come out of plants, they are all able, I believe that they have antioxidant, anti-carcinogenic, anti-mutagenic, anti-inflammatory effects, all these good things. And I think their primary role is to pick up hydrogen atoms from NADP and NADPH, from NADH and NADPH in the cytoplasm and deliver them to the mitochondrial intramembrane space. Quercetin is a popular nutrient used in alternative medicine. Naturopaths like to use quercetin as a plant pigment, and it has there's many wonderful foods that are good sources of quercetin. It inhibits PI3K, which promotes proliferation in cancer. So it induces mitophagy, apoptosis, and protective autophagy. Conclusion, deuterium is an overlooked factor in human health. The body has devised clever ways to manage deuterium, but glyphosate and other chemicals interfere with those mechanisms. Excess deuterium in mitochondria inhibits oxidative stress, induces oxidative stress. Mitochondrial dysfunction is a major driver behind many diseases, including cancer. Insufficient sulfate in the glycocalyx leads to high deuterium in fluid water, where enzymatic action takes place that foils deuterium depletion. Fatty acid metabolism is important for maintaining DDW in the mitochondria. Eating a rich variety of certified organic whole foods is a good strategy for minimizing glyphosate exposure and avoiding cancer. Thank you.
All right, thank you so much for that wonderful presentation. It is now our lunch break time, but we have an amazing sponsored lecture that I encourage everybody to listen to while they are grabbing a bite to eat. It is on the use of fucoidan on reducing toxicities from chemotherapy in the integrative oncology setting. And that is by Dr. Funakoshi from, and this is sponsored by Chimera Functional Foods. And so we are very excited to have this information available to you. And so I encourage you all to join. Have a great little lunch break.